Hello and welcome to this video about amino acid titration. We will talk about amino acid titration curves and how to identify them, how to draw them, and we will solve some problems. So let's get started. I got here the titration curve of glycine. It's an nonpolar amino acid. The titration curve is the same, almost the same for all nonpolar amino acids like isoleucine, leucine, valine, and others. Okay, the beginning point of it. Um, okay, at first, know that amino acids, for titrating amino acids, we start with an acidic solution. Okay, if you add an amino acid in a solution in water, it won't turn the pH down to 2.34 or below that. So we add HCl. We add HCl to bring it down to a certain point. This point can be calculated, but I won't show you the calculation because انتوا بيعجبكم مشاقين. Okay. um, the starting point is here. Grab a pen. لا أخضر. Okay. As we are adding NaOH, the curve, the pH rises. Notice here that there is a plateau because here we have buffering system. Then after it, after we add uh, one equivalence of NaOH, the pH will rise sharply, somewhat sharply, then again another buffering point, then sharply again. These 2 points are where the pH equals the pKa's of glycine. The glycine has 2 pKa's as a diprotic acid. Why? Because it has two dissociable groups. This COOH, which can dissociate to COO minus, and this NH3 plus, it can dissociate to NH2. Okay, remember that we have here uh, the acidic solution. So this glycine in its most protonated form. Okay, it is where the COOH is having an H and the NH2 is having an additional H, which makes it NH3 plus. Okay, as we are adding the base, we are losing protons. The first one that will lose its proton will be the COOH because it is the most acidic group. The most acidic groups are the alpha groups, the alpha carboxyl groups. And even if you have a side chain like glutamic acid has a carboxylic acid in its side chain, it is weaker than the alpha group. Remember this. Also for bases, when they are in the side chain, they are stronger than the alpha amino group. So always the side chains loses electrons earlier than alpha groups. Okay, so at the beginning we have in the protonated form, all the amino acid in the protonated form, we are adding NaOH. At one equivalent, all the COOH will be deprotonated. It will lose its proton because OH minus will steal its proton. And NH3 plus will still have that proton in it. Continuing the reaction, we will move to the second buffering point where we have, okay, I forgot to say that at this point and at this point we have equal concentration of an acid and its conjugate base. I discussed that in the previous video. However, if you didn't see it, okay, at here we have equal concentration of an acid and its conjugate base. What's the acid here? The acid is this, and what's the conjugate base? It's this, okay? So, and when we have this equal concentration, this is where the buffering is at its best. And now, since this glycine behaves as a diprotic acid, it's not a diprotic acid, it's not an acid. Okay, behaves as a diprotic acid. it has two buffering regions. Okay, this the first pKa and the second pKa. If it had like three pKa's, it will have an additional buffering region, which is represented by an additional repetition of this part, like this: chick 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 chick. Okay. Now let's move down and draw some of them, okay, so you understand it more. Then we will solve some problem as we are drawing. Okay, the first one we will draw is that of glutamic acid. Glutamic acid has three pKa's. The first one for COOH, the next one for NH3, and these alphas. These are alphas. And the last one is for the side chain, the carboxylic acid in the side chain. Okay, we have the values. Let's draw it. At first, let us draw the inflection points, which are here. The first one here, the second one here, the third one will be here. Okay, the first one at 2.19, 2.19 around here. The second one will be at, okay, at 4.25 of the side chain. 4.25, 1, 2, 3, 4.25, like here. Shouldn't be exact or accurate, just an approximation. 9.67, 9.789, around here. Okay, in the color to red. Okay, it will start around here in the buffering region. And here it will increase like this, then another buffering region. This like should be more like this. Okay, this buffering region, another increase, and like this, and here like this. Okay, let's get our problems. Okay, the first problem would be the the PI, the isoelectric point. Okay, it's common to ask about isoelectric points. Okay, a question will ask you, what is the isoelectric point of glutamic acid? You draw the titration curve and search for it. Where is it? Okay, we should know where it has its charges. So let's draw glutamic acid. Believe me, this is glutamic acid. It has COOH, it has NH3, and its side chain has another COOH. Okay, it shouldn't be the exact structure of it. This can work. Okay, the first one will be this one to lose. We always lose alphas before. Not always, there is an exception that I will talk about it. Okay, but for now it is always we lose alphas first. So this one will be lost. The first, okay, we always lose acids before than bases. It's, uh, it's common sense, okay? So this one will be lost first. At here, so okay, at here it is this structure, NH3 plus, okay? Here, this COOH will lose an H, so we will have this structure here, okay? NH plus, okay, 3 plus COOH, and here COO minus. Okay, here at this point, at the next equivalence point, this one will lose. Always acids lose before than bases. Okay, so or you can look at here actually and see this here as we are adding. This one is the most acidic, so it will lose first. Then this one less acidic than this one. Okay. The NH3, this value refers to the conjugate acid. Okay, NH3 is a base. Okay, it has a pKb that's around 4 here, 4.5 something. Okay, it's for the conjugate acid. Okay, NH3, NH3, COO minus, and another COO minus, minus. This is plus. And for here, we just copy this and make it NH 2. Okay, and COO minus COO minus. You got it. Okay. The isoelectric point is where we have zero net charge. So we have here one positive charge, 0, 0. Here we have one positive charge, one negative charge, so a total of 0. This is the isoelectric point. Let me color it in blue. Okay, this is the isoelectric point. So you say the isoelectric point is between this value we have here and this value we have here. Okay, it's between this and this. 2.19 plus 4.25 divided by 2 and find it. I won't calculate it. And for all amino acids, you can calculate the isoelectric point, not for all of them. There are also two exceptions. I'll talk about them later. Okay, for most of them, you can calculate the isoelectric point by summing or taking the average of the two closest values. So 4.25 and 2.19 are the two closest values. They are away from 9.67. Okay, for here the 9.06 and the 10.54 are the two closest values, so we take the average of them and we find the isoelectric point. Okay, now let's move to this one, which is lysine. I'll draw it in blue. I already have blue. Okay, 2.16, it's almost the same value here. I'll draw it a little bit higher just to not to having them overlap. The next value will be 9.06, 9.06, 789 around here, and 10.54. It is just a little bit higher right here. Okay, another color, this color. Okay, starting again from here, we'll have the same general shape. Okay, like this. And here, a little increase like this, and like this. This is the titration curve for lysine. So again, if we make the structure for it, we will have here, okay, this, it has NH3, it has an amino group as its side chain, COOH, and COOH is the most protonated form at the beginning because we are in the acidic environment. As we are increasing, now we have here this one, the acid dissociated. This one, this is the acid. Okay, I'm drawing to the left now. I don't know why. Okay, COO minus NH3 plus, 3 plus, and here another NH3 plus. Here we will have the COO minus again, and the first, remember, first alpha groups dissociate first, so this one would be NH3 plus because it is not the most, it is the most acidic, and this is less acidic or more. Okay, this is the most basic and this is less basic, so it is more acidic, so it tends to lose earlier, so NH2. Okay, and here we have the equivalence, the isoelectric point, because plus and minus equilibrate each other and it will have a net charge of 0. Okay, and here I forgot to draw here for this glutamic acid. Here this one will dissociate, so it will be COO minus NH2 2 and NH2. Okay, it will have a negative charge. Okay, the problem we will have here is okay, let's Okay, I'll take that problem later. Let's take, okay, an additional question. Let's say that a question says you have an acid, okay, let me draw it, okay, an acid that has a pKa of 4. Find the, okay, and you put it in a pH equals 6. Find the ratio between the acid and the conjugate base, okay, the ratio between these two. Okay, to find it, and these questions are common in pharmacology because they ask about um, what pH, is it the acidic pH or the basic pH, something or this drug or this molecule will be best absorbed or best excreted by the kidney. Okay, the best absorbed is at when the ratio is toward the non-charged one, and it is the best excreted when it is toward the charged one because membranes are nonpolar, are hydrophobic, they are lipids, so they do not absorb what's having a charge. So to solve this question, we solve it always in the Henderson-Hasselbalch equation. Okay, the equation says 6, which is pH equals the pKa, which is 4, plus logarithm of that value of the ratio. Always the ratio is the base over the acid. Okay, to solve it, 6 minus 4 is 2. Okay, equals logarithm to the 10 for x. So to find x, 10 to the 2, which is 100. So the ratio between the base to the conjugate acid is 100. So this acid, so we are having 100 base to 1 acid. And the base is the negatively charged form. So this in this pH for this acid, it is charged. Okay, it's having a negative charge. Okay, let me write that the acid is HA. Okay, to A minus. We are having 100 HA to 1 for every 1 A minus. And to go back here to the question asks what's the ratio between, let's say, this form of lysine and this form of lysine. Okay, this is lysine, this form of lysine and this form of lysine at pH equals 1.5. Okay, you know at what pH equals 1.5? At pH equals this one, okay, 9.06. You know that when pH equals pKa, the ratio is 1 to 1. So at here, exactly, it's asking here, okay? Without solving anything, the ratio will be 1 to 1. We are having this and this in a ratio of 1 to 1. Whereas if it is asked here, let's say that it asks at pH equals 8, not 8, okay, 8.5, no, just 8, okay. At pH equals 8, at pH equals 8, what is the ratio between this form and this form? Okay, take for the pKa value, take the closest one to this, or the one between these two forms, okay, or the one to the closest pH for the closest buffering region. In this case, we find 8, okay, like this. This is the closest buffering region. If it asks here, we go here. This is the closest buffering region, but actually here it is closer to the equivalence point, so we can say that most of it will be this form. Anyway, let's calculate this one. pH equals 8, and the ratio here, and what the ratio? The pKa here is 9.06. Okay, pKa equals 9.06. put them in Henderson-Hasselbalch equation and find the ratio between them and answer the question. Okay, now let's go to the exceptions I talk about. I said that there are two exceptions, two general exceptions, which are cysteine and tyrosine. Okay, cysteine and tyrosine are grouped in the polar group of amino acids that have a titration curve similar to nonpolar amino acids, except for these two, okay, cysteine and tyrosine. These two, cysteine and tyrosine, have two dissociable groups in their side chain, which are OH for tyrosine and SH for cysteine. Okay, these two can lose their H, so they act as acids, but their acidity is very weak. Okay, so they can lose protons. For cysteine, it's 8.33. For tyrosine, it's around 10 point something. Okay, so they can lose uh, their H plus, their proton. But just keep in mind, this doesn't mean that these are strong bases or weak bases, because here, this value, okay, although these values are large, but this value is for the acid and this value is for the conjugate base. Okay, don't mix them up. So the side chain of cysteine is a weak acid, is very weakly acidic compared to the alpha chain. So drawing the titration curve for cysteine will look like this basic one. Okay. The exception, which I talked about, is that the first one to lose will be this COOH, as always, this alpha COOH. Then this one, because it is the next one in numbers, it is in sequence. They lose in sequence from the most acidic to the most basic. Okay, this one will lose, then this one will lose, and this one will lose. I said that always the alpha will lose first, but here alpha COOH will lose alpha acidic, then the alpha basic, then the side chain acidic. Okay, although this is an acid, but it is very weak, so it will lose later, even later than the base. Okay, to draw it, okay, let me just draw another axis like this. Okay, I will just approximate it. Okay, we are having value right here for the COOH, another value for here around 8, I think. Yes, 8.18, and another value of 8.33, which is here. Okay, it's an approximation. Okay, like this, go up, and here. Okay, this is more like this. Okay, a buffering region, a little bit increase, a slight increase that you can, I don't think that you can notice it in a titration curve, then goes up again. Okay, and ends it. Or you can just add this part that nobody draws it, just forget it. So this is the titration curve of cysteine and tyrosine. Okay, tyrosine will have this one slightly higher. So they look like the titration curve looks like it's a base. Okay. And the second exception I talked about is that to find the pi, the isoelectric point for any amino acid, just take the two closest values and average them. Here it doesn't work because the pi value will be here, which is between this one and this one, the average of these two ones, because at first it will have its Na. Okay, let me draw it. Okay, here the NH3 plus and COO COOH, okay, all protonated, and SH. Here it will lose this H, so it is COO minus NH3 plus and SH. Okay, here it will have C. Okay, it will have COO minus NH2 SH, and here it will have COO all minuses or all deprotonated. Okay. Okay. I wish everything is clear. Good luck in your study and best wishes.